Welcome to the Cannabis Cultivation and Science Podcast. I'm your host, Tad Hussey of KISS Organics. This is the podcast where we discuss the cutting edge of growing from a science-based perspective and draw on top experts from around the industry to share their wisdom and knowledge. My guest this week is Dr. Jordan Zager. Jordan is an expert in natural product biosynthesis and chemical analysis of cannabis resin compounds. Dr. Zager earned his PhD from Washington State University, where his research set the stage for studying the regulatory events governing gene expression in cannabis and their effect on the biosynthesis of trichome-bound cannabinoids and terpenoids. Early in his career, Jordan set his sights on aggressively applying modern scientific methods to the medicinal and recreational cannabis industry and to bring cultivators the tools to become exceptional. Dr. Zager is now the CEO and co-founder of Dewey Scientific in Pullman, Washington. Now on to the show. Hi, Jordan. Thanks for coming on the show today.
Hey, Ted. Happy to be here. Thanks for having me.
Yeah, let's start off talking a little bit about your background. Can you share a little bit about how you got into this type of research?
Yeah, so my formal educational background, I'm a biochemist who chose to study plant biochemistry. I have a bachelor's of biochemistry and molecular biology from the University of Nevada. While I was there, I worked in a plant biofuels lab where we were looking at a potential crop for biodiesel production, specifically looking at the oil quality of the seeds produced by this crop. And it was during that time, as my college years, I found this love for biotechnology. I was a college student smoking weed. And sort of had this idea of let's merge these two things. Let's do some biotech in cannabis. So that led me up to Washington State University in 2014. Recreational cannabis had just become legalized, and they had a market at that time. And I found a lab that studied terpene biosynthesis in glandular trichomes. So at the time, I knew enough about cannabis to know that was very relevant. And just to sort of back up, right? Glandular trichomes are these little tiny cellular chemical factories that stand on the leaf of plants. They're found on about 30% of all plant species. So the lab I entered for my doctorate studied the metabolism in those, and typically these structures, these trichomes, they make essential oil compounds, which for the most part are mono and sesquiterpenes. And now those are famous in cannabis because they provide each variety or strain with their own unique aroma. And, it's gaining traction that it's also these terpene compounds sort of influence the user experience from strain to strain. So yeah, that's how I got into all of this. I eventually was able to work with cannabis genomics as part of my PhD dissertation. And upon graduating with my doctorate in plant biochemistry, went off and started Dewey Scientific, a cannabis-focused biotechnology company where we are trying to make cannabis better through credible science.
And so that's what you're doing now with Dewey Scientific?
That's right. So yeah, at Dewey Scientific, we are implementing modern methods in plant breeding, right? Leveraging the genome as much as we can to identify markers that associate with certain traits, and then make sure these traits get into the varieties that we develop and ultimately put onto the market, both for other farmers to use. And we also have a commercial brand, Dewey Cannabis Company. We're located in Washington State, where we get to sort of market test the varieties that come out of our breeding program and get consumer feedback directly. That's what we're doing at Dewey.
Great. Well, can you just touch briefly on, so what you're doing is breeding, you're not necessarily doing any GMO stuff at this point. Is that correct?
Yeah, that's correct. We are not directly putting genes from other organisms into cannabis. We are not, using various viral or bacterial constructs to sort of manipulate cannabis genetics. All we are doing is, sequencing the varieties we like, sequencing the genome of the varieties we like to sort of better understand what we have. And then once we know what we have, we can start to make targeted and intelligent breeding decisions, right? If you think of the way that cannabis has been bred and treated over the last 40, 50 years through prohibition, right? Breeders have been very limited in their access to technology, their access to facilities. And so it's sort of fallen behind other horticultural crops, right? If you look at blueberry, there's 1,000 papers on blueberry resistance to various pathogens, fungus, insects, and because of the prohibitory nature of cannabis, we just no one's really quite gotten there. So I think that's really what we're focused on, is just catching it up to these other horticultural species from an understanding, and then putting that understanding into practice through smart breeding.
Yeah, I think that's a really good analogy that you're using there, comparing it with vegetable crops that we're more familiar with, because I know when I go out to buy, say, an apple tree, I'm going to look for what diseases are most prevalent in my area, and then look for a particular variety that I know is going to provide some resistance to that. We have a lot of issues with rust around here, for example, with pears. So I know that there's varieties I can choose that are going to be more resistant. I'm going to have a better crop. And what you're getting in terms of a flavor profile too, and yield profile. So I think it's wonderful. So you're just identifying what particular markers may be most useful for someone, and then by identifying, you can then breed plants that have these traits. Is that sort of what you're getting at there?
Absolutely, yeah. So, without the genetic marker component of this, the breeder is sort of left to uncovering things visually as the plant matures. Now, cannabis is a relatively quickly maturing plant, compared to, say, an apple tree. So, the time burden there, an 8-week flower cycle is, relatively quick, but there's so much ground that we have to make up in cannabis that we need to be making decisions a lot quicker. And that's what genetic markers enable, right? So if we now have a genetic marker for, say, powdery mildew resistance, and we want to make sure that this gets into all of our commercial varieties. So at the seedling stage, we can just take a small little leaf sample, run a quick PCR assay, and then we know which in this population is powdery mildew resistant, and then we can get rid of everything that doesn't have powdery mildew resistance, and then we're at ease that all these varieties that we're growing and developing will be protected against powdery mildew.
So it's just another tool, essentially, utilizing technology to speed up the process, because normally, traditionally as a breeder, you'd have to grow out all these plants. You then have to evaluate them, qualitatively in terms of terpenes and the effects of whatever effects that may synergistically occur with that particular plant, how resistant it is to different diseases that it may have been exposed to, and then you may choose to clone or reproduce it that way. But this just gives you a lot more information earlier on if you can identify these markers. And what you're saying is that we haven't identified all these markers, so we don't know where on the genome, powdery mildew resistance, which is what we're going to talk about today, was located. Is that sort of what you're saying there?
Yeah, exactly. We're developing the tools rooted in molecular biology to accelerate plant breeding. Exactly.
Okay, well, let's dive into the paper. And I have to admit, breeding is not, it's not my strength. It's not an area that I have a lot of knowledge in. So I'm probably going to be asking the types of questions to help other listeners like myself that just don't have anywhere near your academic background on this. But the paper itself, I will have a link to directly in the podcast. It's called Discovery and Genetic Mapping of PM1, a powdery mildew resistance gene in cannabis sativa. And this was a paper produced by Dewey Scientific in conjunction with Oregon CBD, and we'll have links directly to the paper. And is there anything else you want to say on that before we dive into the paper?
Yeah, I just want to sort of make clear, I'm not an author on this paper. This was work performed by my company, Dewey Scientific, in collaboration with Oregon CBD, like you had mentioned. But I really just want to give a shout out to Paul Mihalov and Andrea Garfinkel, the two authors. This was something that was a side project in terms of, publishing a public access research paper. I think most companies, if they'd come across a marker like this, they'd seal their lips and it's only theirs. But for us to advance the cannabis industry, we really want to be developing tools that anybody can use. And Paul and Andrea just really took the initiative on getting this work out there, putting it in the public domain, because the cannabis industry is only going to advance if everybody can advance, and if we're holding this close to our chest, we're the only ones benefiting. And, we're just not at that stage in cannabis where there should be a dominant market player. So just, wanted to praise Paul and Andrea, because this is really their brainchild, but they did a tremendous work here.
I appreciate you saying that, and I 100% agree. I wasn't confident in pronouncing their last name, so thank you for giving them a shout out for this work. And the fact that you are sharing this, I think that is important, and something that I hope more companies will do so we can further our overall knowledge. I think that's definitely important. So let's dive into this paper. And again, this is open access, so you can read the paper yourself. We'll have a link on the podcast page. But let's just start off talking a little bit about powdery mildew in general. So what is powdery mildew?
Yeah, so powdery mildew is a common term for a lot of, or I shouldn't say a lot, but of many pathogenic fungi. These are, fungus that attack the plant as a pathogen, meaning it will, maybe act as a parasite, maybe just wants to take advantage of the plant to spread its spores so that the fungus can live another generation. But powdery mildew is one of these pathogenic fungi. And it's been found on all sorts of different crops, of course, cannabis, of course, its cousin hops. It's a problem in the wine industry. It's a problem anywhere where you have fluctuating humidity during the growth cycle of any crop. And specifically, at least in terms of our research on this, we are referring to the specific fungus Golovinomyces ambrosiae, which is what we found on our plants. And we did some genetic sequencing and found that it matches the general term of powdery mildew found in Kentucky and Ohio and Oregon. So it is just a general fungus that attacks crops as they grow.
And I just want to clarify too that when you see PM on different plants, that's not necessarily able to cross all plant species. So we're not going to necessarily see the powdery mildew that we may find on our squash is the same species that's going to be infecting our cannabis. Just something I wanted to point out for listeners.
Yeah, that's absolutely correct.
And since it is a catch-all, we know that there's multiple species that infect cannabis. Is it necessary, really, from a grower perspective, to actually identify the species, or can we treat these all fairly, synonymously in terms of our approach to treatment?
That's a great question, and I don't have a particularly solid answer for that. I think some more research needs to go into this. However, we do know that the species we identified here in Washington is problematic in Oregon, Kentucky, and Ohio. But that doesn't mean that there are others in those locales that other species of powdery mildew that might be inhibiting crop growth. And we haven't done these sort of artificial inoculations of these other powdery mildew species, mostly because of sort of the biohazard that poses, bringing that into a cannabis facility.
Sure. And you touched a little bit on humidity fluctuation. What conditions lead to higher spore pressure and actual infection of the leaf tissue? Yeah,
so things that where the fungus likes to produce more spores and sort of rapidly take over a population, are where you have, larger fluctuations in humidity. Doesn't matter if it's, through the day period or through the night period or in between the two, but it's generally when there's this swing, say you're getting up above 60 to 70% relative humidity and then dropping down to, say below 40. So what that allows is for the spores to be spread when the humidity is low. And when that humidity comes back up, the fungus can then begin to grow on wherever those spores landed. I think some simple ways for people to get around this is just to have a stable humidity in their grow environment. What we've seen, and this is a sample size of one, but at our facility, when we kept things, in the between 50 and 60, we saw a rather large reduction in powdery mildew. So it's really that swing that allows the spores to spread, and then the increase that then allows the fungus to flourish.
Yeah, I will comment just anecdotally. Whenever growing giant pumpkins for years, I noticed whenever I had a really heavy rainy day followed by a super sunny day, that was sort of that trigger. Those big swings was when I would see powdery mildew start to appear in my garden outdoors. And obviously it's something we can't control outdoors, but indoors that's a big thing to control in terms of the environment. And I guess that leads into genetics then. So what was your experimental design, and how did you discover what genetics to really start your project with?
Yeah, this was sort of a circumstance of luck. We at Dewey, we have a very nice plant research facility, and we have these really old growth chambers that they sort of look like commercial refrigerators from like the 60s. Just these big monster units. They do a good job of regulating light and temperature, but they sort of suck with humidity. They don't exchange a lot of air with the external environment. And so we were just housing some of the genetics that we were working on. We had some, a couple dozen different varieties. And it was in our early days. We were leaving these chamber doors closed. Humidity was getting well up into the 80s. When the lights were off, and when the lights turned on, it dried out quite a bit. And we noticed one of our varieties, this PNW 39, which commercially we call Rain Dance, was powdery mildew was not growing on it. So our breeder Paul sort of said, "Hey, I think we might have a resistant variety." So what he did was he took this PNW 39, very resistant. He crossed it with this jumping jack variety that was very susceptible, or on the higher end of susceptibility, and made a breeding cross. Some other background on PNW 39 and jumping jack. PNW 39, while it was susceptible, is a very low yielder commercially. Its THC content is generally sub 20%, so it's not really a commercially appealing variety for farmers. On the flip side, jumping jack is a very high yielder. It regularly produces flower that tests above 28% THC, and that's sort of the farmer's dream. However, it succumbed to powdery mildew whenever it came into contact with it. So what Paul did was he crossed these two. He raised, I think it was just shy of 100 of those progeny, those children. And we took those children and put them into that swampy, humid growth chamber that we have. And we started scoring for powdery mildew resistance. And what we saw, and this is sort of jumping back into high school biology with Mendel, was a segregation pattern of 1 to 2 to 1, which is just classic Mendelian segregation, as it's called, right? Think back to Punnett squares.
Can you elaborate on that for those of us that may not have taken these courses or really remember all of the stuff? I mean, I took a little bit on Mendel, but I've promptly forgotten most of it. So so Gregor Mendel was I believe a Belgian monk in the late 1800s who was
breeding peas, and what he saw was, I crossed a green with a yellow, and some of those kids are green, some of those kids are yellow. And he did it with color and with wrinkled peas. But it pretty much breaks down to, for every gene in a genome, there are 2 copies of it. And in some cases, the genes are dominant. In some cases, they're recessive. And it's possible for any 2 copies to, one be dominant and one be recessive. And so when you make a cross, let's say you have a parent that is dominant with something that is recessive, all of those progeny are going to be what we call heterozygous, meaning it has a dominant allele and also has the recessive allele. However, when you're dealing with things that are heterozygous, when you go to make additional crosses, say with something that's completely recessive or something that's completely dominant, you start to get different ratios of those children. So what we witnessed was just sort of a classic example of a gene segregating in a dominant recessive fashion. That sort of clarifies things.
Yeah, that brings back some some memories there. Yeah, that does make sense. So basically things played out the way you would expect them to from your crosses. And then, so you took those crosses, and what did that research show you?
So what that showed us was that, just because the pattern of the progeny just gave us, okay, there is a single dominant R gene. So that sort of confers what we call vertical resistance to the pathogen that we're working with. And this is something that's been found in, it's pretty common in domesticated crops that you find this single dominant resistance gene. And so that sort of, Paul looked at the data and said, okay, we should do some sequencing here because we can find a marker that will associate with this dominant powdery mildew resistant gene.
So basically what you're saying is this gave you the confidence that you could actually find a particular marker based on the cross that you did. Is that essentially what you're saying?
That's correct.
Okay.
Yep.
So you discovered this marker, which is essentially what the paper is about. What does the research show you? When you guys use the word resistance, for example, in the paper, what does that actually mean? Like, let's just say visually, and then how do you, I know you talk about this in the paper, but how do you show that this plant is indeed resistant?
Yeah, so, sort of to identify powdery mildew on a crop, it has these white little hairs that grow all over the leaves. It sort of looks like you took a powdered sugar shaker and just put it all over the leaves. That's how you can identify it once it's already started to colonize its host. So yeah, visually, we just had this set of children from our breeding cross, and almost every other one had powdery mildew growing on it or had no powdery mildew. It was just leaves. There was none of that powdered sugar on it. So what we did was, we took some of that powdery mildew off. We examined it under a microscope, and that's sort of the classic way of identifying a pathogen, is to look at it under the microscope. You go back and reference various textbooks or resources, and so you identify the spores and you identify the condium is sort of the scientific word, but sort of the legs growing off of the spores, which is sort of how the fungus grows. And we then took a step further. We took this isolated powdery mildew at this point, and we did a quick PCR test and was able to confirm that it was this specific species of powdery mildew.
And then basically all of the plants that had that weren't showing any powdery mildew all had contained that marker, and the ones that did have PM did not contain that marker?
Yeah, so this is where we went and did some, it's called a Sniff Probe. It's Sniff FNP single nucleotide polymorphism. If you've ever taken a 23andMe or Ancestry DNA test, your DNA was run on a Sniff chip. And so what this does is it identifies single mutations throughout the entire genome. So there are cannabis-specific Sniff chips, and we worked with Lighthouse Genomics out of Canada for this specific project. And so we ran DNA samples from all of these children, in addition to the parents, and just looked for mutations throughout the population. And so what we saw with, I think there was 40,000 markers that were run, but on one of the markers we saw, okay, in the parent, in PNW39, the resistant parent, there was this mutation at this specific marker. In jumping jack, the susceptible parent, there was a different mutation at that marker. And then we saw that the children split into three groups, where they had the profile that matched the resistant parent, the profile that matched the susceptible parent, and then things in between that had, and this goes back to those alleles, each copy, where they had one of each of the mutation at that genetic marker. And so then we took that data, we looked back at sort of our phenotyping data, what we saw with our eyes, and we were able to determine that, okay, this is a dominant trait, meaning that it's either dominant or recessive. If it's dominant, it's going to show in the presence of the recessive. And so we saw this pattern where some of the progeny went clustered with the resistant parent that had the same mutation profile at that SNP. We saw things that had in the middle, and then things that went with the susceptible.
So was this a spectrum of resistance that you saw based on how much of that original PNW marker was there?
With this particular population, we did not see a range of resistance, just sort of indicating that this particular marker infers strong resistance. I'm not going to say that's going to be the case for every powdery mildew resistance marker that might be out there. Right, this is just PM1. There's, our geneticists believe there's probably 5 or 6 in cannabis, and those remain to be found. However, with PM1, if you have the correct mutation in that spot, what we saw is that you have full resistance to this strain of powdery mildew.
So what you're saying is there could be down the road other species or strains of PM that could still potentially infect a plant with this marker, but what you saw in your experiment was essentially full immunity in your growth chambers.
Yes, that's correct. I think it's very likely that there are other powdery mildew strains out there or species that could infect a plant that is resistant to the powdery mildew species that we were working with.
Okay, that makes sense. So what kind of conclusions can we draw? So what's the next logical step in regards to this research and advancement? So obviously for you guys, I would assume now you're doing breeding projects where you're going to check for that marker as you breed more of, progeny from this plant line, creating, varietals then that I assume you would then take to market or potentially sell down the road. What's the game plan there? And then how can other growers outside of your company use this research or companies to further advance our overall knowledge in cannabis?
Yeah, so some of the steps that we've taken, the next steps we've taken with this project is, of course, we want this in all of our varieties that we put into our commercial brand that we then sell to other farmers as well. So at this time, I believe we have just about a dozen varieties that we have introduced this powdery mildew resistance into, and those are available for Washington growers. And the other step that we're taking is we want to find these other PM genes, right? It's sort of a dream that we start PM resistance genes, PM 2, 3, 4, 5, if there's that many, and start to stack these resistance genes into a variety. So like you mentioned earlier, right, is it possible for a farmer to have one of these varieties and grow them and still have powdery mildew resistance? If we can get multiple resistance genes stacked into a single variety, it just lowers the chance that powdery mildew can infect the plant, no matter where it's grown, no matter what species of powdery mildew it's subject to.
And that just gets me thinking, do you have in line to do any research regarding botrytis? I know that's another major one that a lot of cannabis growers battle, even more so than PM. Is there that potential there, I guess?
That's definitely not a focus of ours. I think, unfortunately, it is definitely, we are fortunate to be in this situation in general, but, for the sake of botrytis, we're in Eastern Washington, botrytis isn't as big of a threat to cannabis out here. It's a bit drier than, say, the Willamette Valley of Oregon, or anywhere in Western Washington, or even Humboldt, the Emerald Triangle in California, right? As they get fall rains, that increases the pressure of botrytis. So for us in Eastern Washington, it's not as big of a problem. And on the other side of that, we don't want to intentionally bring botrytis into our facility. So we are working with a few folks that may or may not have botrytis resistance. They think they do. So we work with folks on sort of trying to find this marker. And then sort of, jumping back to your question on, how can other farmers make use of this today? What we did was develop a PCR assay, and we provide the primers for that. So, of course, you might need a molecular biologist on your team to sort of run this assay, but we did provide the framework for anybody to be testing for this at their facility if they have the right tools and staff.
So I realize there's some limitations with state, boundaries and such, but if you were a Washington farmer, for example, could they send a sample to you to test for that marker? Is that a service that you guys offer?
That's not a service that we offer, although if there's any interested listeners out there, we are very happy to work with folks on ensuring that consumers are getting a safer product in the end.
And, but are you also, and again, this is something I haven't looked up yet, do you guys offer actual plants then for sale that contain that marker?
Yeah, we do. We can only work within the confines of Washington State at the moment, but we are very happy to work with folks on licensing our varieties. This past grow season, we worked with a large group in Central Washington, getting them some of our varieties, and this is what we do. We license plant varieties to interested farmers.
Great, and that kind of just brings me to a couple more PM questions, if that's okay. One that comes to mind is, have you seen any research that actually shows any health risks associated with consuming or combusting cannabis leaf that has some, level of powdery mildew on it? I wasn't able to find anything online. I was just curious if you had anything on that.
Yeah, I'm unaware of any research that shows that powdery mildew consumption or inhalation is dangerous to one's health. I'm not a medical doctor, but I think it's reasonable to think that it could elicit an immune response. Sometimes that could be in the form of asthma, or it could be a allergic reaction to the respiratory tract. But I think what's more important here is that when powdery mildew is on plants, it reduces the vigor of the plant, ultimately impacting yield, and in the case of cannabis, impacting potency. And that's not something that farmers want to have to deal with. So oftentimes they will treat their plants with damaging fungicides and spray that on the plants. And that's my primary concern. And that's poses a huge risk to the health and safety of cannabis consumers is, are they inhaling combusted fungicides and pesticides? And that's really our goal with this, is to just reduce dependence on fungicides that are damaging to human health.
Absolutely. And I just want to make clear, I'm not condoning the use of or the sale or smoking or consumption of plants that have PM on them. I would assume that any mold spores in general are probably things we want to keep out of our body. But I was just curious if you'd actually found any research. And then in regards to the fungicides, it's an unfortunate reality that some people are still using things like Eagle 20, systemic fungicides and pesticides. So I think that's a wonderful point that you bring up there in that regard. As I understand it, any oil-based or even water itself will kill PM. It just won't keep it from coming back and reinfecting the plant. And you've already elicited the stress response at that point from the plant. So I assume it's more susceptible even than it was before you killed it. So it's something you're going to be continually fighting. Would that be accurate?
Yeah, I think that's accurate. There are other strategies out there other than innate plant immunity to powdery mildew, but it just sort of goes back to maybe it is an organic oil that you're spraying on your plants that is keeping the powdery mildew at bay. However, I mean you're still, when you're doing that, you are adulterating the end product. Maybe let's say you're using neem oil. It's pretty widely used in the cannabis industry as a safe pesticide, but not everybody reacts well to neem oil. I mean, it is a mixture of essential oils, and sometimes essential oils can elicit a negative immune response in people. So while it is much better than using something as dangerous as Eagle 20, it's not going to remove your problem, and it does adulterate your end product.
Yeah, and I would just add that realistically, we don't want to spray anything once the plant starts producing trichome. So once we get a couple weeks into flower, you're pretty limited in what you are able to spray on a plant. My preference, I'll just throw this out here, would probably be something like Suff Oil X or mineral oil based. Even though it's petroleum based, there's a lot more research to support that these products are safer and they don't leave a residue on the leaf tissue, more so even than neem. But yeah, I hear what you're saying. The less we have to spray, not only is that a labor savings, it's keeping us from having to adulterate the plant at all or potentially risk anything being on the leaf surface come harvest time. A lot of people ask me, is powdery mildew systemic? And I don't know if that's something you want to address. I can kind of share my thoughts, but I was kind of curious, how you would describe the life cycle of this set of fungus.
So sorry, your question is it systemic, and what is the general life cycle?
Yeah, that's sort of what a lot of people are asking regarding this fungus.
Yeah, I would say it doesn't. It's only systemic if the environment in which you're maintaining clones or whatever is sort of inundated with powdery mildew spores. And what is the life cycle? This is a fungus. Generally, fungi have two phases of their life. They have their spore producing phase, and they have their sort of fruiting body production phase. How this works is spores are introduced to an environment where they can grow, and they'll start producing. It sort of looks like plant roots. These hyphae, as they're called, through whatever that medium is. Sometimes it's soil, sometimes it's something that's particularly carbohydrate rich, like say a rotting apple, or an apple, it's rotting because the fungus is eating all the sugar. And once it gets to a point where the hyphae, or this, the fungus equivalent to roots, is strong enough, it will start to produce its fruiting bodies. So in the case of mushrooms, right, it's the mushroom. It comes out of the hyphae, it has this fruiting head, and then that fruiting head is actually what then produces the spores for the next cycle. And so you'll see that sort of as mushrooms that have gills underneath, right? Sort of your typical cremini or various mushrooms. They'll drop those spores out of those gills, and then they get spread by the wind or by animals or insects, and the life cycle continues. So in the case of cannabis and powdery mildew, those spores can be dormant for quite some time, and when the conditions are ripe, they can take over a population. So I wouldn't say it's systemic, right? If you take a clone and bring it to a clean environment, there's a moderate risk that plant could then become inundated with powdery mildew, but only in the right conditions. So I think, having a clean facility, especially where you do your cloning, is very important in not passing powdery mildew on from generation to generation.
Yeah, I was just you essentially better explained my thoughts on it, I guess. It's not systemic, but it can act in ways that are similar to something that would be systemic, in the sense that the spores are dormant. They may not be visually present to the naked eye. So when you are cloning, you can, while it's not living inside the leaf tissue, the fungus itself does penetrate the leaf tissue. And so you just want to be very careful when you are cloning, if you are cloning a plant that's had PM, to really do a good job of spraying or treating it prior to cloning, and then even after cloning, just to make sure that you're not creating a situation where you're going to have spore pressure down the road.
Yeah, exactly.
Well, awesome. Hey, I really appreciate you taking the time today to explain this research. Was there anything else that you wanted to share regarding this paper, Dewey Scientific, things that you guys have going on down the road, or future research?
Yeah, future research, and I did mention that we're on the press, or we're looking for other powdery mildew genes, and we do think we have a couple other in hand right now. We're awaiting sequencing confirmation to see that. But I think my big takeaway here is, and I didn't even really touch on this, but none of this would have been possible if it weren't for sort of the collaborative framework that we had with Oregon CBD. I think because of cannabis's history, people are a little hesitant to work with others. But look, none of this would have been possible without them. We had the germplasm, we had some of the key competencies to getting here, and then Oregon CBD was able to complement us in ways that we couldn't do in-house. And I think this is just a testament to what's possible when folks in the cannabis community work together. We can achieve great things, and we're I'm a huge proponent of collaboration and sort of helping other people improve their practice, and hopefully that leads to some more really cool discoveries like what we found with Oregon CBD.
Awesome. Yeah, I really appreciate both your companies doing this work and furthering advancing our knowledge on the subject, and then sharing that publicly, and you taking the time today to come on the podcast. I really appreciate it. And again, just for folks interested, we'll have this available on the pod. We'll have a link right on the podcast page. I highly suggest people check it out. I just want to say thank you again for coming on the show today.
Yeah, thank you, Tad. Happy to be here. Big fan of the show.
All right.
Even before you asked me to come on.
All right. Well, thanks so much. That was Dr. Jordan Zager, and you are listening to the Cannabis Cultivation and Science Podcast. I'm your host, Tad Hussey. I'll post pertinent links on the podcast page at www.kisorganics.com. Just click on the Learn tab and then Podcast. If you're looking for consulting, soils, or soil amendments, or fabric beds or blue mats for your facility or garden, please give us a call or hit us up through our website contact page or tad@kisorganics.com. Thanks for listening.